All right, looks like our recording has started, so we'll go ahead and begin. Hello, everyone. This is Rebecca Dallin with the City of Oklahoma City Stormwater Quality and COSWA, the Central Oklahoma Stormwater Alliance. I'd like to officially welcome everyone to this special training event. If anyone would like to receive a certificate of attendance for today's session, please send me an email to request it, especially if your full name doesn't appear in chat and in Teams. So I will try to send certificates to everyone in attendance, but again, if your full name doesn't appear as your login name for Teams, be sure to send an email to request that certificate. And I'll go ahead and put my email address into chat right now. Please do keep your selves muted during the presentation. If you have any questions, type them into chat and I'll be monitoring that and let our guest speakers know if they have any questions today. We do have 3 guest presentations today from local university students within their fields of study. We have a fairly tight schedule for the presentations and we'll only be able to answer as many questions as we have time for. Additional questions should be referred to their coordinating professors, which is Dr. Jason Vogel for the University of Oklahoma and Dr. Jamie Schussler for Oklahoma State University. A recording of today's event will be available on the COSWA YouTube channel, and I'll post a link to that at the end of today's program. Up first will be Matthew Varriale. He will be presenting field investigations for optimized pollutant removal using orifice underdrains and industrial byproduct underdrain socks. Matthew is currently pursuing a master's degree in environmental science and works as a graduate research assistant for the Oklahoma Water Survey at the University of Oklahoma. His current research efforts are focused on improving stormwater quality using low-impact development and green infrastructure practices such as bioretention cells. Our second presentation today will be given by Jacob McDoulett. He will be presenting Oklahoma Water Survey Bacteria Monitoring Update. Jake is an undergraduate student studying environmental engineering. He's also a research assistant at the Oklahoma Water Survey, and he's the student lead on their fecal indicator bacteria monitoring project. The third presentation this afternoon will be given by Abby Winrich. She will be presenting a hawk's eye view modeling recurrent turbidity impairments and best management practices in Oklahoma. Abby is currently a graduate research assistant pursuing a master's in environmental engineering at Oklahoma State University. Her research focuses on the use of hydrologic models, erosion and sediment, and agricultural best management practices. Prior to OSU, she received her bachelor's in environmental sciences with a concentration in ecological design and engineering from the University of Vermont, where she first developed her passion for water resources. After graduating, she went on to serve as a sustainable agriculture volunteer with Peace Corps Senegal. Since then, Abby has worked on a number of different national water quality projects and hopes to return to work for the federal government after graduation. Well, thank you all for being willing to share your research and information. I neglected to advance my slides, so let me just put the timeline up here real quick. And with that, please take it away, Matt. I will stop sharing.
Okay, thank you. Share first. All right, you can still see the slides?
Yes, that's perfect.
All right, perfect then. Yep, as Rebecca said, I'm Matt Varriale, and I'm currently a graduate student at the University of Oklahoma. And today I want to talk about field investigations of optimized pollutant removal using orifice underdrains and industrial byproduct underdrain socks. And this work is currently contributing towards my master's degree. So for a bit of background, Bioretention cells are a low impact development / green infrastructure practice that can passively improve stormwater quality using natural processes. And so there are different modifications that we can add to bioretention cells that can improve their performance. One of them being adding a orifice to the underdrain of a bioretention cell to help the cell drain slower and increase the retention time. Typically, you're only going to find underdrains in areas where the soil is too compact and can't drain the water out naturally. So, as we all know, in Oklahoma, we have that red clay soils. So, we'll need to install underdrains typically on bioretention cells. Another modification that can be used is adding media amendments to improve treatment. And this can be, there are a couple different processes that can be at play to improve it, such as filtration, chemical adsorption, and storage as well, such as storing it, which will then help foster bioalteration in areas like the rhizosphere and the root structure of the plants and the media. However, using biofiltration media, there are some areas that could be improved upon, such as dissolved metals aren't effectively removed in bioretention. Low sorptive contaminants that don't necessarily adsorb to the soil media, such as neonicotinoids. Some systems have an issue where there's too much organic material, such as composts, which can then lead to actually phosphorus leaching out of the system. And so while, the plants may really enjoy that, that can be quite harmful to the downstream bodies of water if there's too much phosphorus in the system. And then another issue with adding amendments directly to your bioretention media could be that once they're exhausted, you would have to replace that bioretention media, which can, be quite costly and time consuming as well for replacing the whole media system with added amendment. So our solution is, instead of having the amendment in the media itself, we put it down in the underdrain and use it almost like a finisher material, where once the water initially drains through the bioretention media, it will come out the underdrain, where then it will be captured by the amendments to get extra removal or contaminants that weren't necessarily removed in the bioretention cell, such as neonicotinoid insecticides. So this can be a easy, low-cost solution to modifying bioretention cells, and so it can be easier to manage. And so for this project, what we really focused on was making sure that the systems were modular in the underdrains. So each underdrain could be easily swapped out, and each amendment could be easily installed, and just keeping it as simple as possible. So our overall objective is to find the relationship between looking at the orifice size on the underdrain and looking at that industrial byproduct amendment to maximize pollutant removal of biofiltration cells with underdrains in particular. And so what we're even trying to look at is look at different combinations of orifice size, different amendments, and try to target different pollutants as well. So maybe one orifice size and one amendment works best for phosphorus, or another combination works best for neonicotinoids or insecticides. So that's really the main objective of this study. And so our target pollutants are neonicotinoids, which are a cause of concern due to its ability to kill target and non-target insects. One of these non-targets that have ecological ramifications are bees in particular are susceptible. We're also looking at phosphorus. Of course, that's a very common stormwater pollutant, and then metals as well, particularly copper and zinc. And so we first did laboratory column experiments with the underdrain amendments as a proof of concept to make sure that we could actually get, they actually remove what we were targeting. And then once we did that, we then went out into the field and did some field studies. And in this presentation, I will be talking about both of them. So we have our three different amendment types. We have steel slag, biochar, and our fly ash. We chose two different particle sizes. We first have a larger particle size, which is going to be from a 1/2 inch down to a 1/4 inch. And then we have our smaller particle size group, which was 0.0787 inches to a 1/4 inch, and 0.0787 inches. That is a number 10 mesh screen. And so for our biochar, or sorry, our steel slag was obtained from the International Steel Slag Association. They specialize in, this plant in particular specializes in producing rebar out of New Jersey. And what I found interesting about the steel slag was that in the photo, you can see it has a lot of different holes in it and whatnot. And even with those smaller particles, if you were to take a very small particle of the steel slag and put it under a microscope, you would still see those kind of crevices and holes in it, which I thought was interesting. Next up, we'll have our fly ash, which was created by drying out a fly ash slurry and curing it for 30 days. And then once it cured, we broke it up into pieces and then sieved it into its respective particle size distribution. And then lastly, we have our biochar, which which was obtained from a company called Biochar Now. And this biochar was sourced from pine trees, which were then shredded and then underwent pyrolysis at 500 degrees Celsius. And pyrolysis is essentially the process of heating up the material in the absence of oxygen. And so the biochar in particular is very porous material, which has a very high surface area, which can be useful for having more adsorption sites or being able to remove more contaminants. Oh, sorry there. All right, so we have two different laboratory studies which we conducted here. We first have our vertical column experiments, where we first took synthetic stormwater, which which would represent existing water from the bioretention cell. And using peristaltic pumps, we pumped them through vertically from the bottom to the top through the columns with each of the amendments, which then that water would then enter our final point of collection, the sampling bottle, which represents the effluent. And so the other study, which was our horizontal column study, simulates more of a real world practice where the water first enters through a foot of sand vertically and then exits out of the treatment in a horizontal column, simulating an underdrain with each of the three amendments. And so there was 6 inches of ponding at a constant rate, and then 12 inches of sand, and then finally the finishing media. So the results from this study show that the steel slag was the best at removing phosphorus and arsenic, and the biochar was best for removing the neonicotinoids. This is just the horizontal columns, the horizontal column study. The vertical column study also had similar results. And so what the main idea here was from getting these results was to give us a good idea of what to expect when we applied this on a field scale. So our first field experiment was looking at now just the orifice with no amendments. And so this was done at our experimental bioretention cells up in Langley, Oklahoma, at the Grand River Dam Authority's Ecosystems and Education Center, right there by Grand Lake. And so in each of these cells, there was a underdrain, and each underdrain, we had a different orifice size. So we used a 1/4 inch, a 1/2 inch, a 1 inch, and then a 2 inch orifice. Then the 2 inch orifice essentially represented just an open pipe. And so we did this by simulating stormwater events by using water that was pumped from Grand Lake and then spiking it with our neonicotinoids, phosphorus, and metals. And so our target dose rate, pumping it using a peristaltic pump, was 0.1 milligrams per liter. And for this experiment, we did a full hydrologic mass balance on the system. So we collected samples on the initial inflow in the forebay, and then once the water traveled down through the cells, we collected them on the outflow as well when they're coming out of the orifices, and then finally on the overflow as well. So the 2 inch orifice had no overflow whatsoever because all the water was able to just drain through, and the 1/4 inch orifice though had significantly more overflow because you are restricting that flow quite a bit, and so the water will just naturally go over the overflow. And we sampled that as well. And so all of our samples were then composited based on the flow that they represent. And so it's when you're doing a full hydrologic mass balance, it's a lot of samples. And so our preliminary results indicate that the 2-inch orifice works best for maximizing mass removal across the board when looking at each of our 5 target contaminants. And so this then also gave us an idea of what to expect when we started combining both of these ideas. So our next set of experiments was now looking at taking an orifice and combining that with our different amendments. So this photo here shows our typical under drain. And so that was filled with an amendment. And we used our 3 different amendments, like the laboratory experiment, the steel slag, the biochar, and the fly ash. And it's the same idea with spiking it with the rainwater from Grand Lake. And so the main difference here, though, was that we only treated this system as a horizontal column, essentially. So it was a proof of concept of looking at it on a full field context. And this was also with the 1/4-inch orifice. And so essentially we were looking at just the inflow, as you can see that sampling tube on the inflow in the photo, and then the outflow, which was the 1/4-inch orifice. And so for this set of experiments, we sampled at timed intervals. So each inflow and outflow sample were taken 10 minutes apart. And so you would take the inflow sample, wait 5 minutes, then take the outflow sample, wait another 5 minutes, and then take the inflow sample again. And so it was very controlled, very on time with sampling. And so this was the results show here that they were very similar to our laboratory experiments, and that fly ash, or except this time fly ash worked best for phosphorus and zinc. However, biochar still worked the best for our neonicotinoids. And so now that we had an idea of what a full pipe of material looked like with a 1/4-inch orifice, we wanted to try installing 1-inch diameter socks in each of the under drains with keeping that 1/4-inch orifice. And so this would essentially allow us to keep it a little bit more modular. So then the socks were much easier to switch in and out, and it keeps the material kind of in one place instead of just sitting in the under drain. And with the 1/4-inch orifice, the thought was there would be enough retention time to allow those sorption sites to be filled in the material. And so to maximize the removal of the stormwater using the amendments. And so this had the same sampling procedure as before, where we're only looking at the inflow outflow, and that very rigid sampling procedure. And so these results indicate that there just wasn't enough removal going on, even with the 1/4-inch orifice. Using the 1-inch, there just was not enough surface area on the amendments to allow that removal to happen. And so then we went back to the drawing board, and this time went back to using a full pipe full of material. This time we wanted to use a 1/2-inch orifice. And so these field studies indicated that the removal was much more promising. Again, it was only the underdrain. It was not the entire system. And so having that full pipe really does allow for significantly more removal of the neonicotinoids and the metals uh, from the stormwater. And one thing that we also found interesting was that with the 5-inch orifice and the half-inch orifice and the full pipe, we were realizing that the half-inch orifice wasn't necessarily wasn't restricting the flow. It was actually the material in the pipe itself that was restricting the flow. So now it's really a balance between looking at the orifice and looking at how much material is in the pipe, because we now know that the pipe or the material, when it's completely full, will restrict that flow, essentially acting like an orifice. And so that is currently what our current research efforts are focused on. We now know that since the material is the restricting factor, we're doing no orifice and just the material to optimize that mass removal of pollutants. So for our next set of experiments here, we now want to go back to looking at the system as a whole. So looking at the inflow, the overflow, and then also looking at the inflow and the outflow as well. And so we're currently doing that. We actually just finished up an experiment where we did that. And so we're also wanting to look at now looking at different amendments in series. So different removal mechanisms if you have or if you have steel slag followed by biochar or vice versa, and see if that affects the removal, if it improves it or not as well. So in addition to this ongoing work, we are currently looking to work with the city of Oklahoma City at their Memorial Park bioretention cell, which is currently leaching phosphorus. And so we're looking to retrofit that underdrain with steel slag to see if we can get additional removal and try to see if we can remedy that phosphorus leaching. And so, yeah, I'd just like to acknowledge Oklahoma EPSCoR for funding this project, as well as the Oklahoma Water Survey for their continued help. And then, of course, the Grand River Dam Authority for their help with the experiments at Grand Lake. And then also TMS International for the steel slag and biochar now for the biochar. So, yeah, I went pretty fast, so we have plenty of time for questions.
Yeah, great. I had one question. Do you plan to study combinations of media, not just in series?
Currently, no, we don't. And I think one issue with that is the steel slag is much more heavier and denser than maybe the biochar, which is lighter. Uh, and so there can be potential issues with the steel slag actually crushing and breaking up the biochar if it was mixed in completely.
Yeah, that makes sense. I'm sorry, Jason, you had a little bit of feedback, so I had to mute you real quick. I'm not sure what was up with that. That was kind of weird. Did you have anything to add, Jason? Sorry, I can't unmute you, Jason. There you go.
Okay. Yeah, no, I'm in the lab, so you might hear the fan in here.
Yeah. I was
I
like,
got a thing going on right after this, but I just did it from in here. But yeah, he got it, so that's good.
Perfect.
Yeah, when you combine them, the iron slag would basically pulverize the biochar.
That makes sense. So cool. Thank you very much. All right. Well, any other questions for Matt? We'll go ahead and just have you email Dr. Vogel directly, and I will have you stop sharing your screen if you would, please,
Okay.
Matthew, and we'll let Jake get his pulled up.
And Rebecca, if I could make one acknowledgement, those laboratory experiments were completed by Kyle Mattingly, who was in that last photo. So we're presenting them as background, but Kyle, that was Kyle's research. I don't know if Matt necessarily acknowledged that during the time, but he did the lab stuff, and then Matt's doing the field stuff.
Okay, cool. Well, thank you. And thanks again, Matthew, for a great presentation. All right, Jake, go ahead, take it away.
Okay, I'm Jake McDoulett. I'm with the Oklahoma Water Survey, and every summer slides are running away. Every summer we sample for E. coli and Enterococcus, and I'm just going to talk to you about a little bit about what we do and some of the data that we got from last year. So the water survey, we started our bacteria monitoring project in 2018, and you can see in the miles of impaired water, impaired streams for E. coli and Enterococcus. And after 2018, the impaired miles are decreasing in part to the monitoring that we're doing. E. coli and Enterococcus are fecal indicator bacteria. That means that they're indicators of the presence of fecal material and associated fecal pathogens. And so E. coli and Enterococcus, like contact with water contaminated with E. coli and Enterococcus, won't directly cause illness in humans, but they have been found to be a good indicator of pathogens that could and would cause harm to humans. Also, a high concentrations of bacteria can lead to things like lower available oxygen, and so it can lead to damage to the health of the environment as well. These sites that we sample from are spread all over the state, and it takes a lot of time and resources to frequently monitor them. And because of that, the some of these sites, before we started sampling in 2018, some of the sites went almost 20 years without having their impairment status updated at all. And when the sites are monitored, some of them are so small, they're like less than 2 feet wide at some parts, and their monitoring happens with pretty large intervals in between them, and changes in the land use around the stream can impact its impairment status. Things like new suburbs, new factories, new farms, that kind of thing can impact the amount of the indicator bacteria that's in the stream. But with infrequent sampling, we don't really have a representative status of these streams. So our primary objective is to do that, is to get that information, update the status of the streams, and in doing that, we can provide the state with information that it needs to make decisions based on human health risks and the possible uses for each of these water sources. At the same time, we can reduce costs by the state not having to create TMDLs and so on for each of these streams. And then obviously we want to keep monitoring, keep updating, and having a representative understanding of the stream conditions, as well as have me come talk to you and others about what we're doing and the potential benefits that it can bring. So when we monitor for these samples, we use the USGS guidelines for collecting. And this picture on the left is us taking a water sample using the IDEX sample bottles. We take those, we put them on ice, and now we bring them to DEQ, and DEQ does our bacteria analysis for us. And in a day or two, they give us back the amount of each indicator bacteria that was found in the sample. This picture on the right, that is me taking a sample near Stillwater following a pretty heavy rain event. And as you can see, the water there it's red. It's really turbid. And so while we sample, we also take water quality parameters like turbidity, DO, pH, conductivity. And using the nearest mesonet site, we get the local weather conditions. We also record observations that we see there at the sampling location, things like algal blooms, cows in the stream. We have a scale from 0 to 4, 0 being not present, 4 being extreme amounts. And we use that to gauge some common observations that we make, things like oil sheen, fish kill, or fecal material, like stream side fecal material from all of the stream cows. For each site to be eligible to be delisted from the 303D, it needs to have at least 10 samples. And each of those samples has to be taken between the recreational season, May 1st to September 30th. So we will start sampling again in about a week or so. After we have 10 samples, we take the geometric mean of each indicator's concentration. And if for E. coli, if that is less than 126 colonies per 100 milliliter, it can be removed from the 303D. That is 33 colonies for enterococci. We from 2018 to 2023, we chose our sites based on the Conservation Commission's rotating basin schedule. And so in 2018, we sampled from group 3 on this map here, and in each subsequent year went group 4, group 5, etc. And you can see the amount of sites that we sample every year, 15 to 25. And this is that same information, but in map form. And it also includes the sites from 2023 that we sampled last year in the Northwest, the red ones with a star. And for each of those, we've been able to delist 46 sites for E. coli, none for Enterococcus. Some recent research at OU found that the IDEX Enteroler method of bacteria analysis can bring false positives from non-human or originated Enterococci species. And so this summer we have a plan to try some alternatives to help reduce the amount of false positives we're getting. But from 2023 specifically, we have 8 of the 24 streams that we sampled are trending towards the passing criteria. Again, none for Enterococcus. I say trending towards because some of these sites we don't yet have 10 samples for. Many we do. So at the beginning of this sampling season, we will first go to the sites that we didn't complete from last year, get the 10 samples that we need. And the E. coli counts that are below the attainment status are highlighted blue. And as you can see, there's none for Enterococci. The counts are all well above the 33 counts per 100 milliliter required. So for this season, we're selecting sites differently. DEQ gave us a list of all the sites across the state that they wish for us to sample from, ordered by priority. There's 43 total sites that they gave us, and we're not going to be able to sample all 40 this summer. We're making routes on what would be the most efficient to get as many sites as we can. And currently we have routes plotted for 25 of these sites. So we'll be sampling at least 25 sites this summer. One of my favorite parts of the project is some of the training opportunities that it gives to students. We've had dozens of students come through the program and help on the sampling trip, help in laboratory analysis, data analysis. I started at the water survey in late 2020, so my first bacteria sampling was the summer of 2021. And I started as the student lead on the project in 2023, and I'll do it again this year. So it really does provide a pretty major opportunity to learn, to get hands-on experience doing environmental sampling, and opportunity to go to conferences like this. And this summer we will have 7 students, including myself, helping on the project. It'll mix between undergrads and graduate students, but we've had postgrads and postdocs also help us on the project. And so this is a estimate of the economic impacts, both from the cost of remediation and potential recreational value brought from delisting some of these sites so that it's safe to swim in. And from the 46 sites that we have removed for E. coli, it's can lead to it's up to $14 million in avoided costs for remediation. Here are some of the references that I used, and I want to say thanks to DEQ. Huge amount of help. Them taking on the bacteria analysis allows us a lot more time to go to more sites. And then, of course, the water survey, everybody that works at the survey, Dr. Vogel and Matt helps on the project. And the Water Resources Board, Conservation Commission, and DEQ for planning assistant and mapping data. Thank you. I'm free for questions. I think I also went fast.
Cool. Well, thanks so much, Jake. Actually, could you type or copy and paste that link into chat? Would you mind doing that? That last one on the end. We have 2 questions. Do you coordinate with any other organizations through the state or with like Blue Thumb for any of your sampling and analysis efforts?
No, not out of now. It's DEQ does all our analysis, and that's the rest of it we do ourselves.
Yeah, cool. And then another question. Derek says he may have missed it, but are some of your sites rural or were some in urban environments?
It's a combination of both. Last year, the sites were all pretty close to the panhandle and were generally pretty rural. This year, some of the sites that we have are in the Oklahoma City area, and some are extremely rural. So it is both.
Dawson asks, do you assess the other parameters using water WQS? I'm sorry, I'm drawing a blank today.
Water quality standards.
Thank you. Sorry, I'm on the hot seat and I can't figure out acronyms today.
I'll answer that one. Mostly it's there for extra information. If some samples look really weird, we can look at some of the other things and see if there's something going on. But we have not done an assessment with any of the other things that we are collecting. That data is out there, but it's not utilized for water quality standard assessment at this time.
All right, well, thanks so much, Jake. I don't see any other questions at this time. So if anyone does come up with any additional questions for Jake, go ahead and send those over to Dr. Vogel, if you would, please. And I've put his email address into chat again. All right, go ahead and stop sharing yours if you would, Jake, and thank you for posting that link into chat. And Abby, are you ready?
Yes.
Excellent. Go ahead and share your screen if you would. See, Dr. Schussler is helping out there. That's great. All right. Thank you so much. Yes, we can see your screen.
All right. Well, Jake's presentation gave us a great primer on what I'm going to talk about. So my research is, well, first, I'm Abby. I'm a master's student at OSU, and I'm in the Schussler Stormwater Lab. My presentation today is going over some of my research on modeling recurrent turbidity impairments, and then the effect we can have with best management practices in Oklahoma. There we go. Okay, so a little overview of the presentation today. I'm going to talk about what recurrent turbidity impairments are, the way I set them up in the models, and then look at the modeled results and the impacts of BMPs. So first, introduction. What are these recurrent turbidity impairments? So the Clean Water Act set up impairments, and there's two sections that primarily concern water body impairments. Section 305B assesses the overall water quality, and those protect the functional uses. So that's like swimming, fishing, the functionality of the water body. And then 303D is concerned with the compliance and the management to that national standard. So this lays out each and every different pollutant, and it covers both point sources and non-point sources. So point source versus non-point source. A lot of you might be familiar with point sources if you're working more in the urban water quality space, but that's just a discrete conveyance, a pipe or a drain, and it's just a very clearly identifiable outlet. Those are usually managed through TMDLs, and you can have a really good idea of what the inputs to that specific drain pipe look like. A non-point source, on the other hand, is defined as anything that's not a point source. These are whole urban areas, roads, agricultural fields, any bare soils. And there are multiple paths for any pollutants from these areas to enter into the waterway. Because there's not one clear defined outlet, you have to manage them through BMPs. And just make sure that the whole watershed is not having pollutants escape rather than looking at the one outlet point. And just to underscore some of the management challenges here, if you wanted to look at what was coming out of this one drain pipe, you could take the one sample right here and manage the pollutant levels. But if you wanted to look at the impact of this road, you could take a sample here, or you could take a sample a little bit down the road and you might get different concentrations. And you could take a sample on the other side of the road and you might see even different things running off because of the way water flows off roads. So then turbidity, the impairment I'm focused on, turbidity is murkiness in the water. It's caused by particulates that are in the water column that don't allow light to pass through. It's really commonly associated with non-point sources because that's where you're having those soils run into waterways. And BMPs can be used to reduce erosion and therefore manage turbidity. And then best management practices, I looked specifically at agricultural best management practices. So these are a little bit different than what you would be deploying in cities or urban areas. But the way that I'm defining them here is structural or operational changes that you can use to improve water quality. These can be things that are proactive, so they're stopping sediment from leaving the site. These are changes like cover crops, terracing, no-till, that prevents soil loss in the first place, or they can be reactive and they can capture the sediment before it's entering those surface waters. And it's important to note that they have to be maintained and continuously kept up year after year, even once turbidity comes into compliance, to remain effective and still have that management impact. So, and then a little motivation, a little bit of project motivation. Section 319 of the Clean Water Act sets aside specific funding for management of impaired watersheds. It can be used for anything from education and technical assistance to cost sharing for some of the more expensive best management practices. This is a very binary system, so when it's impaired, that money for management is there. And then once the water body comes into compliance, it's just fixed and there's no longer that assistance there. So within Oklahoma, there were all of these impairments that were recurrent. We anecdotally knew this, and the money was going into fixing them, but then they would come up for the same impairment a few years later, and that wasn't a very good use of resources. So we wanted to look and see what was going on there. So into methods, how did I do this? First, we had to find the recurrent impairments. Like I was saying, we knew that this was happening within the state, but we didn't know quite how widespread this was, how many different water bodies were having this issue. So I looked through all the 303D lists throughout the study period and looked for that pattern of listing, delisting, and then a relisting a number of years later. Once we found that pattern, we could put it on, we could map it with GIS and see if there was any pattern of impairment spatially throughout the state. See if these were clustered around like the wetter areas or if they were in the lower flow places. But you can see looking at this map here that there really wasn't a strong spatial relationship with any of the recurrent water bodies. So then to look further into it, we had to select good candidates for modeling. This was things like proximity to the USGS stream gauge, so we could make sure that the model was outputting similar stream flows as to what was actually happening. Similar number of impaired river miles, similar ecoregions. We don't want to compare something all the way on the border of Arkansas with something over in the Panhandle. And then similar drainage area, so we could make good comparisons. Once we did that, we narrowed it down to 3 watersheds to dive deeper into. There was no strong spatial pattern with impairment, but there was a strong temporal pattern of impairment. They're all listed and then coming off of that 303D list around the same time. And then by 2020, they're all relisted again. So to look into what was happening here, we used modeling. I specifically used the OK Hawks model, which is a web-based version of the SWAT model. And if you're not familiar with raster-based modeling. That takes a standardized area, a pixel, and then you add your elevation data so that you can get flow path throughout the watershed. You add a land use on top of that to give you coefficients, practice factor, curve number, looking at how the water is going to move on the land, and then you add soil data on top of that, and you get your soil erodibility and all the physical characteristics that affect things like infiltration. We built those models for each of the 3 watersheds and ran baseline scenarios to look at what was happening during the study period. So all the models were created with that topography, land use, and soil data. And then we used the default values for each of those data sets. The only thing that was changed in baseline scenarios is adding grazing to all of the pasture land within these 3 watersheds. It's not currently assumed in the model, but we know that there's a lot of agricultural activity in Oklahoma, and we wanted the models to reflect what was going on there. And then we ran BMP scenarios to see what the effect of these practices could be. So in Stillwater, we ran a BMP on the grassland, and that was exclosure fencing. And we adjusted the parameters to reenact the physical changes that the BMP would provide. So there's no adding in fencing to the model, but we increased the leaf area index and the P factor as if cows weren't there. So there would be more biomass because the cows weren't eating it down, and the P factor would be decreased because you're not seeing those desire paths from cattle. In Turkey, the winter wheat got a no-till BMP, and that was just removing all the tillage operations from the scheduled operations within the model. So there was only planting, fertilizing, and harvesting. And then in Wowoka, we used conservation grazing on all of the pasture and just increased the minimum level of biomass needed on a field before the modeled farmers were allowed to put their cows on it and graze it. So after creating these scenarios, we can look at the results. The initial model runs gave us an idea of what land use was in the watershed, and then the contributions of each of those land uses to soil loss. So you can see that there are a few main contributors in each of the 3 watersheds. There were 5 major contributors that we pulled out for all 3, Wowoka, Turkey, and Stillwater, which were pasture, grassland range, forest, winter wheat, and then riparian forested wetland. Other encompasses every other land use in the watershed. And for Stillwater, it's important to note that that includes all the urban and developed land, which is Stillwater is by far the most urbanized watershed. We also looked at the contributions of sediment by each of those land uses. So in Wowoka, you can see that just over 25% of that land is pasture, but it's contributing over 60% of the soil in that watershed. And then in Turkey, the largest contributor of sediment is winter wheat. And then in Stillwater, they're all very even, and excluding the urban land, the largest contributor was grassland range. But they're all losing different magnitudes of sediment. So in Wowoka, it's by far the most soil loss happening in the watershed. And you can see that pasture just by itself is losing more than double the amount of sediment that Turkey or Stillwater lose over the entire period. And then again, Stillwater is losing just about 40,000 tons of sediment, and 25 is coming from other, and a significant piece of that is that urban land. So we can also look at sediment loading across the 3 watersheds by normalizing it all to tons per hectare and see how the land uses are performing from watershed to watershed. pasture is one of the most erodible land uses in all three watersheds. But Wowoka, where it is contributing a lot of sediment, is the worst. Interestingly, Turkey winter wheat is losing the most sediment, but actually it's performing pretty well. When you compare it to Wowoka or Stillwater, there's just a lot less of that land use there. The model outputs sediment concentrations, but it's impaired for turbidity, which is an NTU standard, which is that light reflectance. So we can look at the modeled concentration and we can put when it was impaired and unimpaired behind it. And you would expect to see higher concentrations when it is impaired, with lower concentrations when the watershed is not impaired. But we're not really seeing that in the baseline runs so far. There's also large variations in the concentrations. But when we model the BMPs, we can see that some BMPs have a really large effect on the theoretical sediment output of the watershed, whereas some are harder to tell. In Stillwater in Turkey, you can't really see a clear effect from these graphs, but you can see that grazing would have a huge effect in Wowoka. And we can also look at this on a smaller scale. We can look at the subbasins within these watersheds and see where the BMPs are having the most impact. Some of the subbasins in Wowoka can be improved up to 75% in sediment concentration in the outlet of that subbasin. In Turkey, some of the subbasins have about a 10% improvement, but there's also subbasins that are seeing more sediment in that outlet concentration with the introduction of no-till. Stillwater, there's not a huge effect one way or the other. So the different BMPs can make a very large impact in some watersheds, but those same BMPs are not making necessarily a large impact in the others. But the soils are different also across each watershed. This is the K factor on the same scale for each of the three watersheds. Right here is Turkey, Stillwater, then Wowoka. I should have put little headers here for you all. But the soils in Turkey are much more likely to be eroded than the soils in Wowoka or Stillwater. You're seeing a lot more of that green low K factor. The soils in Turkey are much more likely to erode with any poor management practices. This could also affect the impairments because, again, these are all being impaired for turbidity, and the type of soil that's eroding could affect the turbidity readings that we're seeing. So I took samples from various stereotypical land from each of the land uses. So in Stillwater, I went and collected a soil sample on some grassland range. I went out past Enid and got a soil sample from a wheat field, and then went and got some soil from a pasture in Wowoka, and then standardized concentrations. I'm going to go read the NTU values and try and connect the modeled output to more real world values and see how that affects the turbidity. These two samples over here, this is Wowoka and Turkey, and they're both at 500 milligrams per liter of sediment in the water. And you can see that Wowoka would read as much more turbid than Turkey would at the same concentration. So what do we know so far? Extending the same practice in each watershed might not necessarily have the same effect. If we reduce the amount of sediment coming off pasture in Wowoka, because there's so much of it, we'll see a lot more reduction in sediment concentration than we would in either of the other two watersheds. And then agriculture is not necessarily the source of this sediment. Winter wheat is a significant portion of Turkey, but but when you look at it on a tons per hectare level, it's not losing as much soil as some of the other land uses in this watershed. And then soil texture is definitely playing a factor. And it's also playing a factor in where those impairments are likely to occur. Different concentrations are producing similarly high turbidities. So up in the 2000s in Wowoka and then under 200 in Stillwater is still flagging that high enough NTU value for impairment listing. And then we need to find the BMPs that are making the biggest difference and extend those to landowners and practitioners. The practices that we're currently extending might not be having the desired effect and reaching our management goals. So we need to find those BMPs that are actually leverage points and extend those as much as we can. Thank you so much for listening, and I will take any questions that you all have.
Very interesting. All right, so we do have one question from Derek. He says, will this information be used to inform existing or proposed watershed-based plans or TMDLs?
That is the hope. I am working with Oklahoma Conservation Commission, and they manage all the 319 money in the state. So these results will be given to them, and hopefully we can show what BMPs are most likely to be making impacts in these watersheds, and they can adjust what they're recommending and what they're using their money on going forward.
Right, and Jason asks, did you calibrate and validate your baseline Hawks model with existing data? And then he's got compounded questions, so I'll stop there so you can answer that part.
All right, so calibration is actually one of the advantages to the Hawks model. Hawks is that web-based version of SWOT, but with the help of Texas A&M, they calibrated all of the watersheds in Oklahoma to the HUC 12 level, which is what I modeled on. This is also a tool that's available to practitioners, so that was another reason that we chose it, but having it be pre-calibrated was another big advantage.
So the next part of his question.
Sorry.
Oh, go ahead.
Abby, I will say one of my students used one of their pre-calibrated things and we tried to recreate it, and she worked on it for 6 months and couldn't get it to work. So I wouldn't necessarily assume that for different scenarios that it's working.
We're looking at it, the USGS gauges too. That's why I have the USGS gauge in the downstream end of each of the watersheds. The model is also continuously being updated. They put out new revisions, and they'll fix little things throughout the model. So things might have changed. I'm not sure what time frame you had your student working in Hawks, but I know that it's continuously being updated and improved.
All right. Well, I'm not sure if we need to ask the rest of his question, but he said, did you modify historical practices in these regions based on existing BMPs, or assume there was no BMPs historically, or a combination?
In the baseline scenario, it uses that land use map from the beginning of the model run. So there's actually a spin up period to the model. So it freezes the land use at 2000 and then moves forward. So we're assuming no change from then. And we know that there were, thousands of individuals making management decisions in the watershed at that time. But we kept everything default to just see what would have happened if everything was kept the same. What sort of environmental factors would be impacting water quality. And then we added that most commonly extended BMP to the land use that was contributing the most to sediment. And that was also implemented in 2000 and then held on the land with no change for the rest of the model run.
Cool, thank you. Does anyone have any additional questions for Abby? Or I think Jake is still online. Unfortunately, Matt had another engagement, so he had to leave a little bit early. But if we have any other questions, since we're a little bit ahead of schedule, open that floor up. Abby, if I could get you to stop sharing, I'm going to put a little survey up real quick. And while y'all are posting any last minute questions into chat, you can also take this survey for those of you that aren't typing. Just a quick survey to get your thoughts on today's webinar. So Matt, Jake, and Abby, thank you again for being willing to share your time, research, and information with us today. If anyone has any questions that weren't answered during the allotted time for the presentations, please be sure to reach out to their coordinating professors, and I have already put their email addresses into chat for you. Also, if anyone would like to receive a certificate of attendance and your name is not on the list as first and last name, or if I miss you, feel free to email me anytime. And I did put my email address into chat as well. And again, thank you for attending. If you can just do that quick survey, that we'd appreciate that. And of course, if you'd like to re-watch any part of this webinar, we're gonna, if I type the right thing, we're going to have this on our YouTube channel shortly. Our busy bees in our outreach division are working away for Earth Month today, so it might be a few days before we get that up onto YouTube, but that's okay. We'll get it out there soon. Well, thank you again for everyone taking your time out of your busy day to join us. I hope everyone has a great day. I don't see any additional questions in chat, so with that, I'll bid everyone adieu, and I hope everyone has a great afternoon.
Thank you.
Thank you.